So today we are talking about the antibiotic, antiseptic, and disinfectants. We are going to talk about this as a major experiment, and also we will finish the antibiotic producers. So you should know that what is the antibiotic and what kind of different antibiotics we have, and what is their role, so how they works actually. So for example, penicillin inhibits cell wall synthesis, and the antibiotic is the product of microorganism, and the beauty of that is it reacts and inhibit the growth of other microbes. So it's produced by some kind of microorganism and react and inhibit the growth of other microorganisms. So it's really interesting. We have different kind of antibiotic, and today we are going to test the effectiveness of certain kind of antibiotics against the gram-positive and gram-negative bacteria using the disc diffusion method. I will explain to you what is the disc diffusion method. We will use the gram-positive and gram-negative bacteria because we wanted to see that how these antibiotics work against gram-positive and gram-negative bacteria, because we know that each bacteria react to antibiotics in a different ways. For example, the gram-negative bacteria, we wanted to see that if they are more susceptible or more resistant to the antibiotic. But there are some advantages and disadvantages for this method. The advantages is it's quick and easy to do, but the disadvantages is not all antibiotics diffuse at the same rate. So it's important to standardize our experiments. The organism that we are going to use is E. coli, gram-negative, Staph aureus, gram-positive. So what you are going to do, each pair of you guys, if you are in class, but now I'm going to do it, you should label and swap 2 TSY plates with E. coli and 2 plates with Staph aureus to get a long growth. I will show you how to do it. So here is the sterile cotton applicator. Before I take it out, I need to label it. Can I have the Sharpie over there?
Sure. The Sharpie on.
Okay, let me just find it. Should be here. Okay. So we have 4 TSY plates, okay? I will label it first. So initial organism section on the agar side. My initial organism, E. coli, E. coli. 2 of them should be E. coli. The 2 others should be Staph aureus. Staph aureus. Okay? And swap with this cotton. So here, take it out. Here is the Staph aureus. I will vortex it first. Even more. And then we need to swap all over the plate. It's not like a streaking. Because we wanted to get a lone growth, we need to swap all over the plate like this. Because we wanted to get a lone growth, okay? Then, this back. Again, the same thing for the other plates. I will tell you what is the reason that we need 2 plates of Staph aureus and 2 plates of E. coli, because we are going to test both antibiotic and antiseptic. We need one plate for antibiotic and the other plate for antiseptic and disinfectant. So again, all over the plate, make sure. Okay. This is about Staph aureus. The next one is E. coli. This is the E. coli. The same thing we should do for E. coli. So we need to swab all over the plate for E. coli. Okay, let it air dry. And the last one again, E. coli. I'm going to do it in both directions because I wanted to make sure that all plate has this bacteria inside. Okay. Then we need to throw them away in a biohazard material. Okay, so we need to let the plate dry completely before proceeding. But what is the procedure? After the plate dry completely with these two different organisms, we need to divide one E. coli plate and one Staph aureus plate into 4 sections, like this. And label them as 4 different antibiotics: penicillin, erythromycin, tetracycline, and chloromethanical. And this is a procedure that I will show you, but the thing is that we need to do it for both E. coli plate and the Staph aureus. So until here, so far, we use 2 plates. What about 2 other plates? We are going to do it for antiseptic and disinfectant. Antiseptic is antimicrobial that can be used on living tissues, but disinfectant is the antimicrobials that we use on inanimate objects. So these kill almost all microorganisms, these disinfectant, but they are too harsh for skin, okay? Like the bleach, I think all of you guys works at least one time with bleach. So it's too harsh for skin. So the procedure for antibiotic and antiseptic and disinfectant is pretty much the same. There's a little bit different for the procedure, which I will go through it. But we are going to test how effective antiseptic and disinfectants are against gram-negative and gram-positive microorganisms by using disc diffusion method. So the procedure again for antiseptic and disinfectant, which I will go through it, but we started for antibiotic. So our plate is completely dried, Staph aureus plate and E. coli. First, we should divide it by 4. Penicillin, erythromycin, tetracycline, chloromethanical. The same thing for this one, PETC, okay? What should we do? This is the erythromycin disc. This is the penicillin disc, tetramycin, and chloromethanical. And also here we have the forceps. We also have where is the ethanol? ETOH, okay? We need to sterilize our
forceps.
Yeah, with ethanol. The reason is because we wanted to make sure that each antibiotic works differently, right? We don't want to mix it up. So, this is the erythromycin. I will grab one erythromycin and put here in the erythromycin. Super easy. The same thing, because I touched the agar, let it cool down for, not cool down, I mean evaporate the ethanol for 5 seconds. Grab another erythromycin and put it here. That's it. The same thing for penicillin. Tetracycline. And chloramphenicol. I just show it as demo, but make sure you need to use the alcohol between each of them, okay? That's it. This is for antibiotic. Now, when we have to incubate it at 30 degree, agar side down, like this way. But the next thing is for antiseptic and disinfectant. So we are done with antibiotic. The only difference for using the antiseptic is here is the antiseptic and disinfectant. So we need to use this filter, which is the plain, and then soak it a little bit. Let me just show the procedure to you. Okay. Using the filter paper disc, which is plain, touch the edge to the liquid, letting capillary action, wet the remainder of the disc, and place the disc flat on the nutrient agar surface and incubate it. Let me just show you. First, sterilize it, okay? Then, divide it. For example, here is for hand sanitizer, hand soap, and then these two. I'm going to use these four, okay? Should write it down: hand soap, hand sanitizer, Veden, iodine, and envirosite. Here is the list of all antiseptic. So you should know that. For example, we use the hand soap, which is the disinfectant, and also we are going to use one antiseptic, isopropanol or Listerine. So here is the Listerine, okay? Instead of, okay, so I can do the same thing for two other plates, use the other disinfectant and antiseptic, and then you will see the result for all of them. Just go with this one. But you have to make sure that this should be exactly the same for gram positive and gram negative. You know the result. Why? Because we wanted to see the difference of susceptibility for against the gram positive and gram negative bacteria. Okay, but the procedure is like this way. I will grab the plain disc. Open the, for example, this one. Like this. Just touch the edge. Okay? Like this. What was that?
Iodine.
Iodine. This is the iodine. Okay? The same thing. Here. Okay? After you are done with this one, for example, we will go with hand sanitizer. We have to sterilize, get a plain disc, just touch. This is hand sanitizer. Here is the hand sanitizer. And doing the same. for the other bacteria, which is the E. coli. Okay. So we have to do it for the rest of these two. Again, the same thing. So I will do it by myself. Don't worry about it. So, and we will incubate it agar side down for 2 days. In the next session, we will see the results. So basically, if the bacteria is susceptible to that antibiotic, antiseptic or disinfectant, we will see what? We will see here the zone of inhibition, right? So the area around antibiotic which indicator organism cannot grow. This is how we are looking for that. We call it zone of inhibition. Make sure the zone of inhibition for antibiotic producer and these two are the same. The concept is the same, but these are two different experiments. This is antibiotic, antiseptic, disinfectant. The other one is antibiotic producer. But the concept of zone of inhibition means that the bacteria cannot grow when the antibiotic, that specific kind of antibiotic, antiseptic or whatever, is present. It cannot grow. We can see the zone of inhibition. Okay.